I'm Edward Zaga, assistant professor in the behavioral neuroscience area of psychology. I wanted to welcome all of you to UC Riverside. First thing I want to do is to introduce my team. These are 5 graduate students that come to us from 3 different departments at UCR, from the neuroscience department, psychology department, and biomedical sciences. This is what we look like under normal times when we're able to gather, and this is what we've been looking like today. So what do we do? So our lab studies, how are we able to ignore distractors in our environment? So what are the cortical mechanisms that allow us to focus on the task at hand rather than be distracted by all the stimuli around us? And the way we study this is we train mice in these simple tasks where they have to respond to target stimuli. In this case, it's a deflection of whiskers on one side of the face, and ignore distractor stimuli, which are contralateral deflections. Mice learn the task really well, and then we can record the neural activity while the mice are performing the task. The 2 methods that we tend to use, one is wide field calcium imaging. Here's the high speed camera here. We can look at the cortical activity broadly throughout cortex while the animal is performing the task. And the second thing that we can do is we can then use electrophysiology to drill down into single units. So with these 16 channel silicon probes, we can record about a dozen units, neurons simultaneously during task performance. And then also add things like optogenetics, where we can stimulate or suppress a cortical region and look at the effects on behavior and physiology. So that is a very brief overview of what we do. You're welcome to check out my website and send me an email if you have any more questions. But again, most importantly, welcome to UCR, and I hope to meet many of you in person.
Okay, so hello everyone. We are the Goya Lab. We are interested in studying sensory perception. And some of the big picture questions that we're really interested in is looking at different aspects of sensory discrimination and trying to find neural mechanisms that link sensory discrimination to decision making. And so this is just, the amazing team in the lab. I just have everyone's pictures up here. As I mentioned, we're looking at sensory discrimination. And the reason why we're doing this is because, pretty much all of our everyday choices and decisions are heavily influenced by sensory input and sort of our sensory environment. And therefore, looking at sensory discrimination becomes very important when we're trying to study perceptual decision making. And what that really means is, as you're sort of going about your everyday lives, every decision that you're making, which coffee shop to go to, or, which course to take, or sort of, what to order from a menu, everything depends on sort of the sensory input that's coming in and how you sort of analyze this sensory input. And so to do that, we use mice as a model system. And we are specifically looking at a mouse model of autism because sensory issues are very big in individuals who have autism. And there is this idea that's out there that perhaps the sensory issues that are out there in individuals with autism spectrum disorder heavily impair their ability to make perceptual decisions. And then we examine what happens at the level of neural circuits by using 2-photon calcium imaging. And this gives us a glimpse into how do the neurons contribute to the types of behaviors that the animals are exhibiting. Spearheaded by this idea that hopefully we can translate this work into the human population. And simultaneously design analogous behaviors for mice and humans. We should hopefully in the future be able to use these neural mechanisms or insights from these neural mechanisms to come up with therapies for humans. Okay, guys, thank you so much. So essentially, all of the new students, when you come in for interviews, hopefully we can chat more and you can learn more about our lab and get a little virtual lab tour as well. So thank you.
Hello, my name is Ed Kojus. I am an associate professor of psychology and behavioral neuroscience at UCR. My research is centered on cortical mechanism of memory and fear behavior. While my laboratory is located in a new state-of-the-art research facility called MRB, or Multidisciplinary Research Building. Our research focus rather on safety learning mechanisms underlying overcoming generalized fear. Clinical research showed that abnormal safety learning is a major symptom of PTSD. Our studies combine multiple techniques, such as mouse genetics, behavior, electrophysiology, optogenetics, computational analytics. We use in vivo calcium imaging that measures fluorescent signals of neural activation in thousands cortical cells in freely moving mice using so-called head-mounted miniscopes. I do not do this work alone, absolutely not. We have a team of very talented group of young, energetic scientists. If you are interested in our research, do not hesitate to contact us. I hope to see you in person one day. Goodbye. Thank you.
Hello. You are watching the Stanley Lab Tour. This is Dr. Glenn Stanley. And this is me, Yvette Gabriella. I'm a graduate student in Dr. Stanley's lab. Our lab is located in the Life Sciences and Psychology building on the West Wing. Our research investigates eating behavior. We examine the neural circuits underpinning hunger, satiation, and weight gain. This is our wet lab. This is where we store most of our chemicals. We mix chemicals over here, we perform perfusions and histology in this lab space. Our lab investigates GABA A and B receptors in the hypothalamus, the lateral septum, and the septohypothalamic nuclei. We also like to have fun while we work, and sometimes that involves dry ice. The end result of our histology are these pretty little blue brain slices that are mounted on glass cover slides. We take these slides and put them under a microscope to examine if the injections ended up at the correct location. This is our fume hood and freezer, and now we are heading over to the surgery room. This is where we do all of our surgeries. We use this stereotaxic device to complete the surgeries. We implant cannulas into the brain, and we use it to inject our subjects with GABA A and GABA B agonists and antagonists. Then we measure food intake and water intake and different kinds of behaviors and weight gain. This is our station where we fix everything that's broken. Now we're heading over to the graduate office. This office is for both graduate and undergraduate students. This is where we have lab meetings and talk about interesting scientific questions or ponder about the meaning of life. We also like to celebrate when we have accomplishments and eat lots of cake. Thank you for watching.
Okay, we are now in the basement of Olmsted Hall. This is where the lab is. The lab space has not yet been renovated, so we'll do some imagining. This is the front entry to the lab, and that will remain like this. This area is going to get cleared out and become a participant waiting area where we see this big map of New York. This wall is going to come down and will be replaced with a glass-walled office for the lab manager. Along the left, there's going to be some modifications. Some walls and doors will be put up to close off some of these spaces to make 4 testing areas. So just imagine that there's a wall with a door here. 2 of the testing spaces already exist. That's one of them. This will be a 3rd testing space, and this is a 4th one. You can see there's a computer and a desk in there, or a table where a computer will be. In the back here will be the ending edge of the lab manager's office, and this back space is going to be reserved as a reading nook, so a chair, comfy lights and stuff like that. Notice that there's a lot of nice natural light up here. Obviously, there's also some old lab equipment that will be cleared out by the time graduate students are here. And then moving to the connected space, this is the office area. So there will be a large conference table here that will be a RA workspace, as well as where we have lab meeting. There'll be bookcases and a projector screen and a whiteboard. Spinning around to that back wall, this will be an area for desks, and as it looks like now, but of course with new updated desks and chairs. Again, look at these really nice windows that are above, and there will be a frosted glass partition that divides the working area from the lab meeting area to give everyone some privacy. So that is the lab. It will look really different once it's renovated and nice and cleaned up.
Okay, I want to tell you about the planned renovations for the lab space. So this animation is just walking us through what it will hopefully look like. Here we're standing in the first room. This is the one that will have the lab manager's office, which we're staring at the moment, as well as testing spaces. Turning to the right, this is going to be where the table will be for lab meeting. That table will likely also be used for RAs to work at. You can see there's a whiteboard and a projector screen. Flipping around to the back of the room, there's a frosted glass partition, and behind that are desks. These are planned for graduate student desks. You'll notice there are nice windows above them where you can get natural light. So just turning back around, you can see there's some extra little desks and things like that. So there's plenty of space for everyone to work. Moving back through into that first space, the one where most data collection will happen. Here we're standing right now in the waiting area. Older adults, people coming in behavioral testing will sit there. Along the left-hand side, there are 4 dedicated testing rooms. Those are an adaptation of the space that's already there. At the very back, there's just an open space with a chair. My vision for that is it's a reading nook, somewhere that you can do some work without needing to sit at your desk. It's also nice because you, again, have that natural light. And then spinning back around, we're going to take a peek inside of one of these little testing rooms. You know, pretty standard. They should be able to fit 2 adults relatively comfortably. And we will head back out into the hallway, the rest of Olmsted. Obviously, the lab will actually have a roof, but that's how it is.
All right, I am standing here in parking lot 6 on Riverside's campus. This is where the psychology department is. And I'm standing here so that I can show you where a couple things are. So this right over here is the wonderful MRI center. We're not going to go in just in case anyone's scanning today. And I want to show you that it is so close to the Department of Psychology, really right next door. This is the building where my office is and where classrooms for most of the classes you'll TAing are located. And then if we walk truly just 1 or 2 minutes down, kind of where I'm pointing here in the distance to Olmsted Hall, that's where the lab space is. So I just want you to see how close and connected the lab, the psychology department and where you'll be running MRI scans are.
Hi, everybody. My name is Professor Larry Rosenblum, and I'd like to welcome you to the Riverside Audiovisual Speech and Audition Laboratory, or as we like to call it, RASL. In RASL, we study speech and talker perception as a multisensory function. Regardless of your level of hearing, everyone lip reads to some degree, and our research shows that integrating audiovisual speech occurs automatically and unconsciously. We found that even pre-linguistic infants integrate speech this way. We also study the unconscious imitation of speech that we all do when we talk with each other. We found that being able to see, as well as hear a talker's speech, increases the amount of imitation we perform. RASL also studies how we all use multisensory information to recognize people. We've discovered that people's voices can be matched to their faces, and that this ability can be based on perceiving a talker's specific manner of speaking. Overall, our research suggests that when it comes to speech and talker recognition, the human brain is designed around multisensory input. We believe that RASL's research can help us better understand and support individuals with sensory impairments. Stop by and say hello the next time you're around.
Welcome to the Laboratory for Cognition and Action in the CCN area of the UCR Psychology Department. I am David Rosenbaum. In our lab, we study how the mind controls body movement in the world. Here's an example of the sort of study we conduct. This we do outside our lab. We have an indoor and kind of an outdoor lab too. This subject shown here started near the yellow tape in the back, way behind her. She looked forward, and like most subjects, she chose to pick up and carry bucket B, which was near her start position, but far from the end, which you're looking at now. She did not pick up and carry bucket A, which was farther from her start position and nearer to the end. That's an odd choice, but almost all people do this. We call this precrastination. It is roughly the opposite of procrastination. It turns out that precrastination occurs commonly in everyday life. We answer emails too soon, pay bills too early, buy in bulk needlessly, submit forms or papers too soon. We carry too much stuff in too few trips, convict people prematurely, have surgery before getting all opinions, and accept or reject options too quickly. This phenomenon has been covered widely, for example, in The Guardian of London and in the New York Times. Our hope is that by studying even elementary actions, we can learn a lot about how people think and act in the world, and hopefully better the lot of all humankind. Thank you for visiting the Laboratory for Cognition and Action.
Adjacent to the psychology building is Olmsted Hall, where many of our labs can be found, including the Laboratory of Aging and Neurocognitive Imaging, or LANI Lab for short. Our space includes 2 offices for graduate students and postdoctoral researchers. There is also an open meeting area for group discussions and lab meetings, as well as birthdays and lab celebrations. Behind the meeting area is a large open work area for the lab manager and undergraduate research assistants to process data. And there are 2 behavioral testing rooms, each set up for administration of neuropsychological and computer-based cognitive tests. You might also find our lab members at the Center for Advanced Neuroimaging, which is on the other side of the psychology building. This is where all of our MRI scans are done. Our research focuses on the intersection of aging, the brain, and cognition, with an emphasis on learning and memory. Typical research questions ask, what is the effect of aging on brain structure and function? What is the effect of aging on performance across a variety of learning and memory tasks? What are the neural substrates of these learning and memory tasks? And does the effect of aging on the brain explain age-related declines in learning and memory? In addition to working hard, we have a great team who likes to play together. Thanks for visiting the Lonnie Lab.
Hi everyone, my name is Annie Ditta and I'm the director of the Teach Lab here at UCR, which stands for Teaching, Engagement, Active Learning, and Creative Thinking in Higher Education. So broadly, I study the scholarship of teaching and learning, which is interested in understanding the effective teaching practices as well as effective learning practices and the intersection between the two so that we can create the best possible learning experience for our students. Some of the questions that I ask in my lab with my lab team include, how do we increase student motivation to learn, particularly when they're not excited to do so, when they're in the class, or they may even be avoiding enrolling in certain classes due to this low motivation? How do we help them get excited to learn? I'm also interested in understanding things like, how do these new practices, like taking photos of lecture slides, for example, how does that influence our memory for information that you're trying to remember for a test or perhaps even more long term? And I'm also interested in understanding the effect of exposure to examples on learning, since there is some research to show that examples are helpful for understanding concepts and perhaps to inspire new ideas, but they can also act as harmful blocks that get us unnecessarily fixated on examples and prevent us from thinking more broadly about how this knowledge can be transferred to new scenarios, which is the ultimate goal of learning. So this is just a brief sampling of some questions I'm interested in, but broadly teaching and learning topics are of interest to my lab group. And so where do we conduct this research? Well, most of it happens in my actual classrooms. So in the lecture halls when I'm teaching in person and it's not COVID time, now in the online teaching space. I also have some lab space. I don't have any photos to show you at the moment, but generally speaking, I am conducting research with the students in my classes live as I try new teaching practices and teach them new learning strategies. So I hope you enjoyed visiting the Teach Lab. I look forward to meeting with you all soon. Bye.
Did you see my hand?
Hi, I'm Rachel Wu, and I'm an assistant professor of psychology at UC Riverside. They seem to be much more confident, less afraid to face any challenges that come their way. They seem to be more outwardly, willing to face anything that comes their way.
I'm John Franchak, and I run the Perception, Action, and Development Lab at UCR. What we're most interested in is how do infants and how do children figure out where to look at the right time. Everything is happening really fast in all different directions, and we can only choose one thing at a time to look at. Infants come into the world not knowing where to look. And so in one of our studies, we use a device called a desk-mounted eye tracker, and that's a monitor that has a special set of cameras on the bottom that record infants' eyes to see exactly where in the video they're looking at from moment to moment. And then all we do is really just show them a bunch of different TV shows and music videos, and we see, do they look at more informative parts of the scene, or do they look at places that are colorful or that have lots of fast motion. And what we found is that with age, infants are increasingly figuring out exactly which parts of a TV show are the most relevant, the most informative places to look. In the other context that we study visual attention, what we really want to know is, how do they look at the real world? To measure eye movements in the real world, we need a device called a head-mounted eye tracker. This is a little bit more tricky to do because it means that we actually have to put a hat on the infants. We have to have them wear a band with a special set of cameras that move around and record their eye no matter which way they're facing. So to calibrate the eye tracker, we show infants a board that has lots of little windows cut out, and we show them toys in each of the windows to figure out what their eye looks like in the camera and how that matches up to where they're looking in the world. And then once we have the eye tracker calibrated, we're able to turn them around and show them different types of objects. So in the study we're doing right now, we show them different pictures of flowers and cups to see how they choose to turn their heads in different directions. We used to assume that what infants see in the world is just like what adults see. But really, infants are on the ground and parents are up there kind of out in their own world. And so the more studies we're able to do, putting head-mounted eye trackers on babies to see how they interact in the home, how they interact in the labs, we're going to learn so much about how infants and children experience the world from their own perspective.
Hi everyone, welcome to the CAN, the Center for Advanced Neuroimaging at UCR. You see that we're right next to the psychology department, 5 minutes away. We have parking right outside for participants and their families. This is the space. We have a testing room right here where families and research participants can come in. They can fill out their consent forms, they can fill out their questionnaires and behavior forms behind a locked door. Workspace with different workstations for staff and students and faculty if they want. We have all of our different software uploaded. You'll see on the right in the back, there's TMS equipment for those of you who are interested in transcranial magnetic stimulation. We can interface that with the scanner. Right, and here's the control room. So what you'll see is stimulus presentation computers where you can also see some diffusion tensor imaging data. We interface with a very sensitive VPIC system that allows us to capture rapidly behavioral responses. It also interfaces with an eye tracking equipment inside the scanner. In addition to that, we have psychophysiological equipment with a BioPack that allows us to measure skin conductance and respiration as participants are responding in the scanner. So if we go right inside here, you'll see that we have a 3T Prisma scanner. This is the first Prisma scanner in Southern California, and it's also the same scanner that's being used by multiple large-scale collaborative projects, including the ABCD study, the Adolescent Brain and Cognitive Development study, the ADNI study, the Alzheimer's Neurological Initiative, and HCP Lifespan. So we've laid out a couple of things here for you just to give you a little flavor of what's all possible, but essentially we have multiple response boxes depending on your task, depending on your paradigm. We have 2-button responses, we have 4-button responses, among others. We also have an EEG cap. For those of you who are interested in EEG, we can interface that with the neuroimaging as well. With two different head coils, a 32-channel head coil, 64-channel head coil. I also mentioned an eye tracker as well. And for those of you interested in animal work, we also have a little rodent scanner here too, and the capacity for rodent neuroimaging as well. Welcome to the KIND Lab, the Kids Interaction and Neurodevelopment Lab. My name is Kolya Michalska, I'm the lab director. And at the KIND Lab, we're interested in how children understand the emotions of other people, mostly fear and distress. So how they understand them, how they react to them, how they remember them. So we're interested in biological differences, temperamental differences, also in the influence of parenting, maternal parenting, paternal parenting, and in broader cultural, contextual influences.
Hi, I'm Dana. My research focuses on how children with anxiety and other forms of psychopathology perceive emotional or other threatening stimuli. And I like to use computational methodology in my research as well. And in my spare time, I like to play tennis, knit, and I'm trying to learn the piano.
Hello, I'm Matthew Kirsting. I'm a first year in the KIND Lab. I'm interested in children's moral development and moral reasoning, as well as their causal inference and how fear and empathy relate to those factors. In my free time, I like to read as well, and I play guitar and play golf, which is very nice in California because it doesn't get cold here.
Hi, I'm Jordan. I'm a third-year graduate student in the KIND Lab. Broadly, my research interests center on children's emotion perception and understanding of threat. And then more specifically, I'm interested in how negative life experiences like discrimination may impact children's understanding of others, as well as whether parental socialization of these experiences may serve as a protective factor. One of my most favorite things about working in the KIND Lab is how close all of the lab members are, from our undergraduate research assistants to our lab manager to the graduate students and then to Kalina. There's just a ton of opportunities for collaboration, and that's something I really appreciate about our lab.
Hi everyone, my name is Nikki. I'm the lab manager at the UCR KIND Lab, and my research interests include the development and treatment of anxiety with the specific interest in how the arts and creativity can be helpful forms of intervention. As far as hobbies, I love to practice yoga, engage in different types of arts and crafts. And my favorite thing about the KIND Lab and Riverside in general would be its people. I think we have a very lovely community of people who are very supportive and kind.
In addition to the grad students and our lab manager, we also have 12 to 15 undergraduate RAs. They're currently working virtually, but here's where they sit when they're here in person. And a beautiful view. So yeah, my favorite thing about the KIND Lab, I really like the environment. We're all really supportive and it's casual, we get along well. My favorite thing about Riverside is to have really, really good Mexican food.
We have a lot of fun, we get a lot of interesting work done, and it's just a great time. And the Riverside area is a beautiful place. It's surrounded by mountains. Right now they're snow-capped, so even driving around, going to do mundane tasks like getting the groceries makes it seem like it's a very majestic place to be.
We have some exciting research studies planned in the years ahead, and we hope to meet you and hope you join us. So stay safe, everybody, and thank you.
So here we have two more KIND Lab members. Hi, Nikki.
Hi, my name is Nikki. I'm the lab manager at the KIND Lab. I help with RA scheduling, interface with families, and just help the lab run smoothly. And my favorite thing about the lab is just the people. We have very nice, kind lab members, so yeah.
Great, thank you. And this is Chris.
Hi everyone, my name is Christopher. I am a first year neuroscience PhD student at UCR, and I'm currently doing a rotation in the KIND Lab. And what I'm looking at is how children remember and store fears, and sort of how that may influence their responses to certain faces and things like that. And another thing I really like about the lab is also the people, and I think the studies that we're doing are super interesting, and I'm very excited to kind of dive into those.
Welcome to the Developmental Transitions Lab. My name is Misaki Natsuaki, and I'm the director of DTL. In our lab, we study children and families to understand how family context and biology matter for child development, including the development of psychopathology, such as depressive symptoms, conduct problems, and anxiety issues. And to do so, we use various types of study designs, including adoption design, sibling design, and twin studies. And we also employ a long-term longitudinal design, which we've been following children since 9 months old and all the way to adolescence. And in my particular interest in research is the transition from childhood to adolescence. And remember that awkward time? Yes, that's the one that I'm particularly interested in. And in particular, I'm focusing on how puberty has effect on the development of depressive symptoms during that time. And I would like my graduate students to introduce themselves. So here's Jin.
Hello, I'm Jing Wang, a fifth-year graduate student in the Developmental Transitions Lab. My research focuses on the synergetic influences of multiple factors in the family system in understanding child development. For example, how infertility experience impacts parenting and marital relationship, which then spill over to child outcomes. Now, let's meet Eric.
Hi, my name is Eric. I am a third-year graduate student here in the Developmental Transitions Laboratory, and my research interests are primarily developmental disabilities as well as bereavement. Now, here is Arash.
Hi, my name is Arash. I'm a first-year PhD student, and my research focuses on psychopathology, especially from a developmental perspective. Specifically, I'm interested in how family dynamics, such as parent-child relationships and other early social experiences, can lead to internalizing problems such as depression.
Welcome to UCR.
Welcome to UCR.
Welcome to UCR.
Welcome to UCR.
Welcome me as I take you on a tour to the YHD lab. So we're located in the Olmsted building, specifically 2139. This is our lab space. Welcome. These are testing stations for our participants. We also have different stations for our RAs. We have a microwave, refrigerator on the other side. This is working space for our RAs. We also use it for storage for our journals and books. This is our first office for our graduate students. As you can see, we have ample window space to see nature. This is another office for our graduate students. Now you'll have the opportunity to meet our advisor, Dr. Lloyd, along with some of our lab members.
Hi everyone, welcome to UC Riverside. My name is Erica Lloyd and I am an associate professor in the developmental area in the Department of Psychology. And I also direct the Youth Health and Development Lab. My research centers around positive youth development and prevention science, particularly for kids of color. And one fun fact about me is that I share the same birthday as Beyonce. I look forward to meeting all of you.
Hi, I'm Marilyn. I'm one of the undergrad RAs at the lab. One of my research interests is identity development in youth, especially within communities of color. And one thing that I love the most about the lab is how much we always try to engage with our community.
Hi, my name is Chelsea McElwee. I am a first-year PhD student in the developmental psych area. I am interested in exploring factors that impact Black and Brown youth from thriving in inner city communities. A fun fact about myself is that I enjoy spending time volunteering at local community-based organizations. I look forward to seeing you around.
Hey there, my name is Tate LeBlanc. I'm a second-year graduate student here in the lab. Broadly, I'm interested in how identity, efficacy, and aspirations develop among Black youth, both in online and offline contexts. When I'm not in the lab, you might find me at the gym or at my desk at home playing online games like League of Legends. I hope you're enjoying the tour so far, and we look forward to meeting you.
Hi everyone, my name is Lanisha Williams, and I am a second-year graduate student in the YHD lab. I'm interested in how early educational experiences influence identity development, and my favorite thing about working in the YHD lab is that everyone is personable, so it makes it a fun place to work.
Wow, that was an adventure. I hope you enjoyed the tour to the YHD lab, meeting some of our lab members and our advisor, Dr. Lloyd. We look forward to seeing you around UCR.
Hi, my name is Jamie.
Hi, my name is Ashley Marin. I am a second year student here in the Childhood Cognition Lab, and I am working on my SYP project, which looks at the role of context, cognition, and social cultural factors on over-imitation.
Hi, my name is Haejung Lee. I'm a fourth year grad student working in the lab, and I currently research how children use their imagination to learn about their social world.
Hi, my name is Jae Sun. I'm the lab coordinator here. I'm interested in learning about how parent-child interaction influences how children learn about the world.
Hi, I'm Rebecca Rickert. I'm the PI of the Childhood Cognition Lab, and we study how children's cognition is influenced by fantasy, media, and religion.
Hi everyone, my name is Diamond Bravo and I'm the principal investigator in the Cultural Resiliency and Motivation Lab. In my lab, we use MEX methodology to investigate how risk and resilience factors simultaneously inform motivation and healthy development among marginalized adolescents and emerging adults of color. Now, I'd like to give you a quick tour of our lab, tell you about some of the projects we have going on, and introduce you to our fabulous graduate students. Our lab is located on the 3rd floor of Olmsted. And this is our newly renovated conference room where we hold lab meetings, collect data, and document days in the lives of first-generation community researchers. Here we have our testing room where research assistants code and analyze data. And this is our research assistant office, a quiet home away from home with a view. And now for a quick peek at 2 ongoing research projects. The first study is an international community partnership study based in Cuba. We use mixed methods to understand the role of community and ethnic, racial, and cultural factors in relation to the well-being and career motivation of medical students in Cuba. So why Cuba, you might be thinking? Well, here's why.
They're known as the Army of White Coats. Today, Cuba has one of the highest number of doctors per capita in the world at 8.2 doctors per 1,000 people. That's 3 times higher than the US. But there's one thing that sets Cuban medical graduates apart: their community focus. Cuba's medical education system exists within the healthcare system. Instead of teaching hospitals, there's teaching at community centers and polyclinics. Polyclinics are community-based clinics with primary care physicians, specialists, and diagnostic testing. And each clinic tailors their medical approach to the needs of that community.
As you can see, the Cuban healthcare system is unique and offers a valuable opportunity to learn how preventative medicine and community-based healthcare might be adapted and applied to reduce disparities in our healthcare system. And the real heart of the project is our community partners in Cuba. The second project I'd like to share is one that examines the cultural elements of Hispanic-serving institutions. This project is in collaboration with the Chicano student programs here at UC Riverside.
Briefly, we want to know how cultural environments affect the well-being of marginalized student communities, particularly those that identify as first generation. And now to wrap us up, our grad students.
My name is Brenda Rincon, and my research interests center on the social support experiences of undergraduate and graduate students of color. I'm really interested in learning more about how support from parents, romantic partners, peers, and mentors affect students in regards to their sense of belonging, academic motivation, experiences with imposter syndrome, and overall health.
My name is Ashley. I am currently a third-year graduate student here in the psych department. My research interests primarily center around people's behavior and productivity as they approach deadlines. And part of how I study that is by investigating procrastination, especially as it's related to motivation, emotion, and affect.
Thank you for watching.
Hello and welcome to the Culture and Child Development Lab. I'm here to give you a brief tour of our lab space. So if we come in here, we have our main lab space, and this is where we have all of our research assistants work on coding or data entry. So normally they would all be seated here. So we have all these computer desks here. And then if we continue through here, we have our desk for our lab manager. So our lab manager usually sits there. And you can see here that we're still decorated for St. Patrick's Day because that happened. It was right before the pandemic happened, so we're a bit frozen in time down here. And then if we continue through here, we have our childcare room. And so this is where we often have mothers and child participants come in when they're going to participate in the study, and we might have them come in here and do the consent and assent forms. Oftentimes, if both the mother and child are involved in the study and there are younger siblings, we will watch them in here while the mother and child are engaged in the study in the other rooms. So this is our childcare room. You can see we have lots of children's books and lots of games and activities. Okay, and then if we continue through here, then we get to our other office spaces. So here we have lots of office spaces for graduate students. Here's one here. This is one of our graduate student offices. And then if we continue here, this is one of our experiment rooms. So this is where we often will have participants when they have to complete an experimental study. So they might be in here completing a problem-solving task or completing a survey. And then if we continue down here, we have a second graduate student office here. So this one is shared by 2 graduate students.
Hi, welcome to the UCR Adversity and Adaptation Lab. I'm Tuppet, and
I'm Bree. I'm Duan. I'm Jessie.
I'm Anna Maria. That's Sandy. Go ahead, last but not least,
Marian.
Yay!
Hello, welcome. My name is Brent Hughes, and I direct the Social Neuroscience Lab at UC Riverside here on planet Earth. We study questions related to how humans think about themselves and other people, how we learn about our social environments, and connect with others. We use methods like behavioral experiments, neuroimaging, computational models, and linguistic and semantic analysis to gain deeper insight into these questions. Our lab members will now tell you a little bit more about our work.
Hi, I'm Eleanor. I'm a fourth-year grad student in the lab.
Hi, I'm Genesis, the lab's research coordinator.
We study social connection and communication.
One interesting thing we found is that certain communication styles may help listeners empathize with speakers better when they're sharing emotional experiences.
For example, it can sometimes be helpful to use more emotional language, more vivid language, and speak faster.
We're excited to continue studying ways that humans can connect with each other, and aliens too.
Hi, my name's Jake. I generally do research on how people think about themselves and the various identities that they belong to, and how those identities and the norms associated with them might obligate people to behave in a particular way or think in a particular way. Some of the research that I've done has found that people kind of infer particular attributes ought to be true of their group if they're true of themselves, and that people might resist updating self-beliefs that have more implications for other self-beliefs.
My name's Irian Dermo, and I study motivated information sampling. Basically, what I'm interested in is how people's group-based identity attachments, be that their political affiliation, their gender, their ethnicity, etc., influence how and where they gather information, and then how they update their beliefs as a function of whether that information favors their beliefs or not. One interesting thing that I found is that people generate greater variability within their in-group experiences, which they then cherry-pick to arrive at favorable conclusions.
Hi, my name's Julia. I'm a graduate student at the Social Neuroscience Lab. I study the other race effect, which is the tendency for people to see out-group members as more similar to one another while seeing in-group members as more distinct. We're currently investigating this through two main methods. In a psychophysiology study, we're measuring how people learn to identify threat to in-group and out-group members and how they then generalize the learned threat to other members of those groups. We're also studying this through a neuroimaging study by analyzing neural adaptation in the fusiform face area after participants see faces of varying similarity, race, and facial emotion.
I'm Jennifer. I am a first-year graduate student, and I'm ultimately interested in the ways in which self and identity relevant information are represented through self-referential cognition, like episodic memory and self-relevant attention. For example, I'm interested in how experiences with social stigma and bias affect the ways in which individuals from marginalized backgrounds represent and perceive their self and identity. I like to examine whether the stability and integrity of self and identity representations are threatened by such negative social information. And eventually, I'd like to apply these developing theories in self-representation stability to individual differences in affective neuroscience in ways that affect low self-esteem, social anxiety, depression, and more.
Hi, I'm Arshia. I'm the lab manager for the Social Neuroscience Lab. I study social connections, especially when our experiences and cultures are different and history is so unknown. Research in psychology has shown that we are not nearly as wise about others as we think we are. So I'm most interested in answering why we so often misread the minds of others and how we can improve. I'm currently testing the concept of emotional granularity, the ability to differentiate specific emotions, and how it relates to how well we understand our own emotions and as well as others.
Hi, I'm Jimmy Calancini, and I'm the principal investigator of the Riverside Social and Spatial Cognition Lab.
Hi, I'm Emily Esposito, and I am a second-year graduate student.
Hi, my name is Liz Wilson, and I am also a second-year graduate student.
Hi, I'm Deja Simon, and I also am a second-year grad student.
Hi, I'm Darian Dick. I'm a fourth-year undergrad and an RA in the lab.
Hi, I'm Tiffany Garcia. I am also a fourth year undergrad and an RA in this lab.
Hi, I'm Hansa Cantor. I'm a third year and an RA in this lab.
So we're social psychologists, and we primarily conduct research related to stereotyping and prejudice in the domain of intergroup relations. Let's take a quick minute to hear what the grad students are working on right now.
So with my current line of research, I am looking at intergroup attitudes and their effects on things such as migration and person-environment fit. Basically, what I'm curious about is if people migrate to places based on their attitudes as well as maybe their identity, such as if they are a member of the LGBT community.
In my work, I apply multinomial processing tree models to different measures of implicit intergroup bias and implicit identity.
And my current research broadly examines whether people better recognize other people, things, etc., that they consider part of their in-group.
All right, I hope you're having a great day here visiting UC Riverside virtually, and looking forward to talking to you soon.
Hello, potential UCR psychology graduate students. I'm Kate Sweeney. I'm a faculty member in the social personality area in our department, and I'm also a member of the faculty who participates in the health and well-being specialized subfield of study. In my lab, we study the psychology of uncertainty, which includes topics on stress and emotion and coping. We also look at these processes in the context of healthcare. So some of my studies are conducted at local hospitals and clinics. And we also run a lot of experimental studies in the lab, plus other field studies online and in other locations. I have a really big lab. At the beginning of next year, I'll have 4 students, 2 of whom are co-advised. And we have about 40 or 50 research assistants that work with us during the year. I am happy to answer any questions you might have about my lab and our activities.
Hello, my name is Sonia Lubomirsky, and I've been doing research on the science of happiness for over 30 years. And primarily, my students and I have been studying what makes happiness shift over time, like both up and down, but mostly up. How do we make people happier? And the most common tool that we use is what we call happiness interventions, which are experiments kind of like clinical trials, where we randomly assign some people to engage in some kind of positive activity or behavior, like expressing gratitude or doing acts of kindness for themselves or acts of kindness for others. And then we measure their happiness over time. And so basically what we're trying to figure out is, how and why does engaging these behaviors make people happier? And after decades of research, I have landed on a conclusion that sounds like a cliche, but it's very well empirically supported, which is that social connection is the key to happiness. So most recently, my students and I have been focused on studying what fosters connection, how do you make people feel connected through, say, engaging more social interactions. We're studying the substance MDMA and how that fosters connection, how having deep conversations and listening well fosters connection. And so it's a really, really great topic. I have a lab of right now 5 PhD students who are all fabulous. One of them can be here today. Her name is Nina Radosic, and she's an international student from Croatia. And she's interested in connection and particularly conversation. So she's studying what fosters feelings of connection and well-being through conversation. And so now we're going to turn to, and I'll have my other 4 students introduce themselves. And we're going to start with Annie.
Hi, I'm Annie Regan. I'm originally from Massachusetts, and broadly I'm interested in studying well-being interventions and the features that make them work or hopefully not backfire. And that's another interest of mine. And currently I'm working on a self-compassion intervention looking at emotion regulation as a potential mediator for well-being benefits.
Hi, I'm Madison. I'm originally from California, so now back here. I'm broadly interested in studying curiosity right now, so I've been really looking at developing a study that examines ways to evoke curiosity and if that could help with things like communication and social connection.
Hi, my name is Tanya. I grew up in Ecuador. I'm broadly interested in how alternative medicine and emerging technologies can mediate well-being. And right now I am working on a study to understand how taking MDMA can help foster feelings of connection through a conversation.
Hi, I'm Ramona. I'm from Irvine, California. I'm interested in volitional personality change, goal pursuit, and self-reflective processes in goal pursuit. Right now, I'm running a study where I ask people to be more extroverted, that's outgoing, assertive, and energetic, and also testing the efficacy of a self-regulation tool to help people to that end, and also testing the upper limits of this intervention and seeing who this works for. Does this work for introverts, for instance, or people who struggle with shyness?
And that's it from us for now. Wishing everyone more happiness.